CAM stands for Crassulacean Acid Metabolism, which is a specialized form of photosynthesis that's found in plants that occupy arid or semi-arid environments. And basically, it's a water-saving type of metabolism that allows the plants to become more water use efficient. CAM plants evolved approximately 30 to 40 million years ago. And as the atmosphere became more arid, there was an additional radiation of species, and many more CAM species became present or evolved on the order of 5 to 10 million years ago. To fully understand Crassulacean acid metabolism, you have to understand that there are 3 major types of photosynthesis in plants. There's C3 photosynthesis, which is the most prominent kind, and that's found in about 90% of all plant species. And a good example of a C3 plant is rice. And then you have what's called C4 photosynthesis. And C4 photosynthesis is found in about 4% of all plant species. And a good example of this is sorghum or maize. And these are highly productive plants because they add an extra step to photosynthesis. And that extra step is that they fix CO2 through an initial carboxylation reaction that fixes CO2 into C4 acids using an enzyme called phosphoenol pyruvate carboxylase, or pepcase for short. That C4 acid is then transported into a bundle sheath cell, and the CO2 is concentrated to reduce the process of photorespiration in the bundle sheath cell. And this acts as a turbocharger for photosynthesis, so it improves the efficiency of photosynthesis. And then finally we come to CAM photosynthesis. And this does the same thing as C4 photosynthesis, but instead of using a division of labor between 2 different cell types, plants fix CO2 primarily at night. And they do this with the same enzyme of PEP carboxylase that C4 plants use. And this is found in about 6 or 7% of all plants. And a good example of that would be pineapple. And the reason that CAM plants take CO2 up at night and use this primary carboxylation reaction with phosphoenol pyruvate carboxylase is that they can open their stomata when the temperatures are cooler and there's more humidity in the air, so the plants lose less water. And then during the following day, what happens is that they take these stored C4 acids and they decarboxylate them. The CO2 is released into the mesophyll cell, similar to what happens in a C4 plant, and the CO2 is refixed by Rubisco in the normal Calvin-Benson cycle of a C3 plant. One of the important things about CAM plants is that they're highly water use efficient. So if you look at the 3 different types of photosynthesis that we discussed, we have C3 photosynthesis in a rice plant, C4 photosynthesis in a maize plant, and then CAM photosynthesis with a cactus. What you'll notice is that if you compare the water use that's consumed by these 3 crops, CAM plants use only 20% of the water that a C3 plant like rice would consume in a year. The modern definition of CAM is a plant that shows nocturnal acidification through the accumulation of these C4 acids, mainly malate. The other definition is that they show net nocturnal CO2 fixation at night. Without those 2 things, you don't have a CAM plant. We talked about the core metabolism of CAM, but CAM plants also have a number of other anatomical traits that are going along with this photosynthesis to make them more water use efficient and more drought tolerant.
CAM is often associated with a number of anatomical traits such as tissue succulence and other water storage organs. Also, thick, waxy cuticle to prevent excessive water loss, lower stomatal densities, and highly responsive stomata which limits water loss. Also, shallow roots to maximize the water uptake of precipitation when it occurs, which is often unpredictable under arid and dry climate conditions. CAM occurs in about 38 plant families and is present in around 6 to 7% of all vascular plant species.
In the bromeliad family, we have Ananas camosus, the pineapple. It is a highly valued fruit economically. In addition to pineapple, we also have bromeliads that are economically important as houseplants.
Another highly valuable species are the agaves, such as Agave tequilana, which is used for the production of tequila, and also Agave americana, which are both used as low water input crops for biofuel production.
In addition to agave, we have here two different cactus species. This is Hylos cereus, and this is the dragon fruit, and it's valued for its fruit. And then this is a member of the cactus pear family. This is Opuntia ficus indica, which is useful for not only its fruit production, but the pads are used as a fresh vegetable. And importantly, this is also used as a biofuel feedstock.
Another important houseplant is Oncidium panamensa, referred to in a common name as dancing ladies. We also have vanilla. This is a vanilla orchid. It will bloom and create a pod which will dry, and that's used in cooking.
Here we have a highly valuable plant, which is aloe vera, with many purposes as cosmetics, pharmaceuticals, nutraceuticals, and also for wound healing and burns in the skin.
This is a pencil plant in the Euphorbia family. It's an important houseplant, and it is an Old World CAM species.
Here we have a selection of Kalanchoe species, which belongs to the Crassulaceae family, from which Crassulacean acid metabolism derives its name.
In addition to the economically important CAM plant species that we've discussed, there are also important CAM species for research purposes. And what I have here is Mesembryanthemum crystallinum. This is the common or crystalline ice plant. And this is a facultative CAM plant, which means when we drought stress it or salt stress it, it will switch from C3 metabolism to CAM. And that switch is very important because it allows us to distinguish which genes are important for CAM photosynthesis. And this is important for our CAM engineering strategies.
We have sequenced ice plant genome in collaboration with Dr. Cushman at the University of Nevada, Reno. First, we isolate DNA from the ice plant. We embark a fascinating journey to assembling its own genome. It's kind of like a jigsaw puzzle, and we use the isolate DNA and assembling into entire genome. The ice plant is facultative genome, which includes C3 and CAM state. To understand that changes and the CAM state, we need to have an entire genome and understanding which gene is the most important. Ice plant genome opens up new avenue to understand its special characteristics and advanced in the CAM research. By employing sequencing technologies and bioinformatics tools, we can connect all the DNA fragments to entire picture of ice plant genome. This complete genome will help us understand about 200 genes known to be associated with CAM, which is related to the signal transduction, transporter function, and metabolism. To identify which gene is important to the CAM state, we do the RNA sequencing analysis over 2 days during the day and night. And to compare C3 and CAM state, we use the bioinformatics technologies, and then we can identify which gene is the most important during the CAM state.
Recall that the CAM plant at night fixes carbon dioxide via bicarbonate into oxaloacetate and then malate, which is then stored at night in the vacuole. If we sample the plant at dawn and then compare the sample at dusk, we should observe a nocturnal acidification by a pH indicator, chlorophenol red. Here, you can notice the difference between dawn and dusk samples.
The carboxylation module, which is active at night, is responsible for fixing carbon dioxide into malate. The process starts when free atmospheric carbon dioxide is converted to carbonate, which is then combined with phosphoenolpyruvate in a carboxylation reaction to produce oxaloacetate. The oxaloacetate is then converted to malate, which is then stored in the vacuole overnight. The daytime active decarboxylation releases carbon dioxide from the vacuolar malate that was stored in the previous night. The resulting pyruvate is then reconverted back to phosphoenolpyruvate to begin the CAM cycle anew. It's thought that the release of CO2 by the decarboxylation module signals stomata to close, which is exactly the opposite of how stomata are regulated in C3 plants, where the stomata are open during the day and closed during the night. Both the carboxylation and decarboxylation modules were built separately, and when we combine them, we obtain a full diel CAM cycle, which represents a fully synthetic reconstruction of CAM metabolism. Arabidopsis plants expressing the carboxylation module show nighttime malate accumulations and are generally larger in size. The plants expressing the decarboxylation module show a general depletion of malate. When we combine the two, we observe nighttime accumulations of malate and daytime depletions of malate, much like you'd expect in a CAM plant. To test the drought resilience of the carboxylation module, decarboxylation module, and the full CAM circuit expressing plants, we withheld water for 18 days and then rewatered the plants for 3 days to assess their survival. The decarboxylation module and full CAM expressing Arabidopsis plants showed significantly higher survival, and we think this is due to the daytime closure of the stomata, which limits transpirational water loss.